Welcome, everybody. So we start. So very briefly, a few words about RareCyte. RareCyte is an American company founded in 2009, and we are based in Seattle. And we are a precision biology company developing high-performance microscopes dedicated to pathology type samples, all type of biopsies, including liquid and tissue biopsies. At RareCyte, we focus on two main areas. First, the rare cell analysis and retrieval of the cells. It's mainly used for the CTC, the circulating tumor cells. And second, the fast-growing field of spatial biology and its highly multiplex tissue analysis. So today I will talk about the Orion, which is a really high-speed, highly multiplex spatial analysis imaging platform. And if some of you are interested, contact us after the webinar, and I will send you a few words about the SiteFinder 2 instrument here on the left, which includes, beside the imaging part, an onboard device for the micro-region retrieval to collect the cells of interest for downstream genomic analysis. So first of all, why did we develop the Orion system? So as you know, the tissue consists of heterogeneous cell types where cells have diverse functions and functional states. The spatial biology goal is to understand which cells are present, where they are localized in the tissue, but also the pattern of biomarkers expression, ideally at the cellular level. The goal is also to understand how the cell organize and how they interact to influence the tissue microenvironment. So resolving this complexity is challenged by several things, challenged by the autofluorescence of the cell, and maybe challenged by the overlaps of the different fluorescent signals, which limit the number of targets that can be simultaneously evaluated. It's also challenged by the very low throughput, usually due to a very long multi-cycle staining procedure, and also by a very slow acquisition time. When we have asked pathologists and researchers to list the characteristics of the optimal tissue imaging platform compatible with pathology workflows, this is the feedback we have received. I'm not going to read now all of this in detail. I just want to highlight here some keywords. We can see 10 or plus simultaneous markers, whole slide, rapidity, high resolution, to have panels and flexible choice, etc. And it's within this frame that we have developed the Orion platform. It includes hardware and software combined with a strategic fluorophore palette to enable rapid, high-resolution, whole-slide spatial biology results via a practical single-cycle process. So from a high level, Orion has a very straightforward workflow. Essentially, you prepare your slide on traditional microscope slides, so the normal tissue section. We are working mostly with FFPE preparation. I think what's important here, it's first, Orion analysis is done on the whole slide. And second, the staining is actually a single-step staining process, which is done with really standard immunofluorescence protocol with all the different biomarkers addressed at the same time. It takes only one scan for all the fluorescent channels, and then the data can be analyzed downstream, either on the onboard qualitative software named Artemis, or on a third-party software. So the question is, how do we do this? So, well, we are still using fluorescence because fluorescence is really needed for the high speed we want to achieve. In short, we image roughly 1 square centimeter, and we can do that in a little bit less than an hour and a half. So it's very fast. Traditional fluorescence imaging, as you know, use a single dye in each spectral region of the entire spectrum, and that, generally speaking, allows for a maximum of 4 or 5 markers to be viewed at the same time. Some people use an alternative approach. Here, represented at the top of the screen. So they use an alternative approach to increase the number of markers. They perform sequential or iterative immunostaining by repeating many cycles of staining, imaging, stripping, washing, and then again the staining, imaging, etc., etc. With the high risk to damage the tissue, with the difficulties to stack the different images acquired at each cycle, and again with the very low throughput associated to this method. But with the Orion technology, here at the bottom of the screen, we extract specific signals from dyes that are closely adjacent in the spectrum, which allows to assess multiple markers per spectral region. So with Orion, we leverage the fluorescence in a smart way. We are doing all the color at once, and then we resolve the spectral overlaps by using a powerful algorithm to perform a deconvolution. To illustrate this, we can take this example. This section of lung is showing 5 key biomarkers, cytokeratin, CD68, and 3 T cell markers. The 5 antibodies used here are conjugated to 5 dyes, which are all from the same region of the spectrum. It's the orange region, so around 600 nanometer. Orion is able to resolve all these signals, so each individual biomarker can be visualized independently. That's what you can see along the bottom of the slide. With no intervention from the user, the cellular complexity becomes resolved. Keep in mind that here, for this example, we have done this for a single spectral region, but now with Orion, you multiply this across the entire spectrum, and so you increase a lot your possibility for multiplexing. We have also optimized some panels. For example, our immuno-oncology 12-plex panel contains the typical immune response marker and also some specific checkpoint markers. And here on the left side, you can see our immunoresponse 9-plex panel, which is giving a more general immune response. Those 2 core panels, they offer some flexibility in order to include custom biomarkers of your choice, such as architectural markers, cancer-specific biomarker, but it could be also marker of the cellular cycle or specific of neuronal disease, for example. As I mentioned earlier, Orion is able to scan the whole slide. So here is an example. This is a 17-plex panel applied on a tonsil section. We have scanned here a little bit more than 1 square centimeter of tissue. It took less than 2 hours to image everything in the 17 channels. The image here is showing the architecture from high level, but of course, Orion is offering very high resolution as well. Most people who use the system, they use our 20x objective, which gives about 0.5 micron resolution. It is possible to get 4x, 10x, 20x, and 40x magnification. Here, it's 20x. You can see down into the different subsets at the high-level resolution over the whole slide. And here we have zoomed in on 5 different areas of this tonsil section. To help with the visualization and the understanding of these 17 biomarker staining, it's easier to look at combinations of given biomarkers. Here we have 4 times the same region which is displayed with 4 different combinations of marker, which we are going into detail in the next slide. As you can already notice, the very good resolution here allows us to really have a look at the phenotype at the single cell level. So here we see a B cell follicle with most of the proliferating B cells located in the germinal center. So there are CD20 plus in blue and PCNA plus, so in red. The CD40 lymphocytes in yellow are mainly confined to the interfollicular T cell zone. As for the macrophages, which are in magenta. They are mainly concentrated within the follicle and are relatively few in the interfollicular T cell zone. On this second image, we confirm with Ki67 in yellow and pCNA in red that the proliferation of the B cells is intense within the germinal center. The PD1 plus cells in light blue, they do not proliferate, which is consistent with the presence of this immune checkpoint inhibitor. And around the CD3 plus lymphocytes are found in the interfollicular zone. Here on this third image, we have zoomed in on the interfollicular T cell zone, and we can see there the expression of FOXP3 in red on one subtype of T lymphocytes, so on the CD4, which are in light blue, but not on the CD8, which are in pink here. I would like to take just a few seconds to remind you that with Orion, we work with antibodies that are directly conjugated. There is no need of whatever strategy of signal amplification, no secondary or even tertiary labeling, no pyramid signal amplification. So using directly conjugated antibodies, Orion enables here to collect a very clear image of an intracellular marker such as FOXP3, for example, which is expressed quite weakly. And this is possible thanks to the use of 7 high-power lasers installed inside the Orion instrument, and thanks to a specific algorithm to treat the signal. Finally, on this last image, we are focusing on the myeloid cells. We see a co-expression of PDL1 and CD68 in red and in yellow on the macrophages of the germinal center. And the M2 macrophages, which are CD163 plus in light blue, are excluded from the germinal center. We can see them actually right here on the last image on the right at the bottom. So with Orion, we can assess any kind of sample, lung, melanoma, tonsil, thymus, pretty much any kind of tissue that you can put on the slide will work well with the Orion system, including tumor microarrays. There is no problem at all to scan this type of sample. Back to another example using the same panel, the 17-plex. Here apply on normal lymph node on the left, on tonsil during inflammation, it's in the middle, and on a chronic lymphocytic leukemia cells in a lymph node on the right. And without going into a detailed analysis, just based on the colors of the different areas, we can already see clear structural differences. On the normal lymph node, so on the left, the B cell follicles, which are in blue, contain few proliferating cells and no germinal center. The follicles are surrounded by T cells areas in white. In the tonsil during inflammation, so the image in the middle, we have many of the B cell follicles that contain germinal centers, those red areas that you see, which is consistent with the inflammatory response. And then for the lymphoma, completely on the right, clearly the proliferating B cells are dominating. There are very few germinal centers and very few T cells. It's actually quite common for people to perform serial sections for H&E staining, the hematoxylin and eosin staining, to look first for specific areas of concern and then look deeper with the immunofluorescence staining. With the Orion workflow, it's actually feasible to get the H&E imaging of the same section. So the same exact slide can be stained. What we do is we first stain the sample for the multiplex fluorescence, do that imaging using the Orion system, remove the cover slip, and then we stain the slide again with H&E and image it again using the bright field mode of the Orion system. So we can see for the same section, the H&E staining, as well as the high-plex fluorescent imaging, as you can see. At this stage, I would like to invite you to visit later our webpage and to play with 2 demo datasets that we are providing. To explore the data, it's easy. Just go to rarecyte.com/Orion and click on View Dataset. The first dataset is a 16-plex image of oral squamous cell carcinoma, and the second is a 17-plex panel on a lung sample. Once you have selected one of the datasets, you will then be able to look at the whole slide scan and to zoom into it as you wish and display the different color corresponding to the 16 or the 17 biomarkers. Regarding the analysis with Orion, we are providing the Artemis software to visualize the image acquired. From Artemis, we can export pyramidal open source format files. So the analysis can be done on many software solutions already existing, such as QPath, Allo, Visioform, but also some open source and free analysis solutions that have been developed by different research groups. Orion data can be integrated into an existing quantitative analysis pipeline without the need to modify the exported file. In this example, Allo AI by Indica Labs is used for the quantitative analysis of images obtained with Orion. 3 kinds of samples were analyzed: normal lymph node, the tonsil during inflammation, and the lymphoma. 2 algorithms were used here. The first one for the segmentation of the cells to separate and measure each individual cells, and the second algorithm to run a tissue classification and recognize automatically some types of tissue like the B cell zone, the T cell zone, the epithelium, and the stroma. And then from that, it's possible to quantify the various complex phenotypes in each of the different areas of the tissue. I can share this poster with you, just ask me. And to give you more information on quantitative data, I would like to highlight a recent publication in Nature Methods, which presents mcmicro, an end-to-end processing pipeline to transform large multi-channel whole slide images into single-cell data. Also, you will see on the right-hand side of the screen a link to a webinar given by Sandro Santagata of Harvard, who highlights the use of this tool, mcmicro, that they have developed to analyze the Orion data. To summarize and close my presentation of the Orion technology, here are 5 points which I believe make Orion really a unique tool. The possibility of doing whole slide imaging will prevent sampling errors, allowing to see actual patient-to-patient variability. The straightforward single tissue staining procedure, which removes experimental bias. The speed of acquisition and the contained cost per slide that enable translational and clinical studies. The possibility to image with true subcellular resolution, enabling real tissue mapping, and then the possibility to image the same section in bright field after standard tissue stains, which is very important for pathology labs. Well, this is the end of this presentation. I hope you are now enthusiastic to join the world of spatial biology using our Orion technology. And we can move now to the Q&A session. Here are our contacts.
Thank you, Mickael. So we have had a few questions coming through the Q&A box. I'm going to be asking you these questions and hoping that you have the right answers for the audience. So the first question that we have here is about the antibody conjugation process, and especially the question is about whether it's possible for a user to conjugate primary antibodies themselves and combine them with existing panels.
Yes, actually, the way we are developing our Strataplex reagent is with Orion. We have a palette of fluorescent molecules that can be conjugated to your own antibodies, to the antibodies of your choice, to include them into the panel. So doesn't mean that it's not possible to use another molecules, but as a good start, I will highly recommend to use those conjugation kits. It's very straightforward procedure to label the antibodies, and then they will fit perfectly with the rest of the panel.
Thank you, Mickael. Another question is about tissue autofluorescence. So is the Orion platform able to handle tissue autofluorescence? And a question related to that one, do we have any pre-treatment recommendation before we do the analysis or the acquisition?
So in our procedure, when we prepare the slide during the staining part, just before doing the staining, we actually have a process to try to decrease a lot the background by doing some illumination of the tissue. So and for the rest of the autofluorescence, it's actually quite useful to have a little bit of autofluorescence of the tissue, because we can visualize that also during in the final image that we have acquired. And so it's the autofluorescence, it's actually, I would say, useful to see the general morphology of the tissue, and so it's always good to have a little bit of it. But yeah, the purpose of the algorithm that we use and the convolution that we perform is actually to make sure to remove all the overlaps between the different fluorescence that we are using, but also we are taking care in the algorithm of the autofluorescence. So there are several steps of making the final image very clear and beautiful.
Thank you, Mickael. Another question that we have is about automated tissue staining and whether we do recommend using an autostainer for sample preparation.
Well, you certainly can use autostainer for Orion samples, but it's not essential. One of the core design features of the Orion is to remove the burden of long multi-step tissue handling procedures by enabling a tissue staining in a single step. So you can either use automated or non-automated sample preparation, whichever suits your existing workflow. But I would say it's not mandatory. It's a very simple procedure, classic procedure for staining. And I would say I do not expect people to have to actually stain 40 samples at the same time with a huge panel. So, but yeah, it's compatible, but not mandatory.
Okay, thank you very much for that. We have another question, but I think you answered it already, about the nature of the tissue specimens that can be used with the Orion technology, mainly if FFPE samples are amenable to the technology.
FFPE, frozen samples, whatever you can put on the slide, I would say, we will be able to scan it with the Orion. It has to be a microscope slide.
Okay.
Classic.
Thank you. Another one is about the objectives. The question is whether we do have the aforementioned objectives already pre-installed in the system, or whether there is a selection that needs to be made before the system ships.
So I would say the instrument come with all the objectives, the 4x, 10x, 20x, and 40x. Now, what is the recommended magnification? For instance, for subcellular localization, you can use the 40x objective. So he has a resolution of 0.2 micron. And then the system also has the 20x objective on board. And this one gives a resolution of 0.5 micron per pixel. So, but he's going to go to scan 4 times faster than the 40x. So you don't have really to give up throughput and resolution, the throughput for the resolution. Short answer, I would say the majority of the users are using the 20x because it gives very good resolution and it goes very fast. So that's, but if we look at the process, the way the machine is dealing with the scan, it will first run a scan at 4x. Then using that very quick scan, we are going to let know the software where we want to make the image. Using a very basic tool, you just tell the system, okay, I want this area to be scanned, and then it will start the scan using the 20x or 40x that you have selected. That's the process.
Okay, thank you, Mickael. Another question is about the menu of panels that we do currently have, especially regarding the mouse model. Do we have anything right now which is ready for mouse specimens?
Short answer, we do not have yet panels for mouse. We have developed 2 panels for human. I expect that the panel, the 9-plex panel, which is the basic immuno answer, the immuno response, will be the one that people are going to use. And based on that very small core panel, it's small. It's already a 9-plex. You can add your antibodies of choice, or you can start from scratch. But I would expect that people always want to, at least in the immuno-oncology field, they will want to measure those markers. So why not start with something with a core panel and just build your personal panel on the top? But for the mouse, we don't have anything available. We don't have a commercial product yet for that.
Okay, thank you, Mike. I think we can take 2 more questions. So another question is about the loading of the slides in the system. People are trying to understand if there is a loader within the system, or if you need to load the slides one by one.
So the procedure to load is very straightforward. You click on open, then you have the 2 holders that go out of the system, and you just manually load your 2 slides, click close, and press scan. So in terms of interaction with the machine, it is very, very simple. There is no other action to do. To answer the question, we do not provide an auto loader or something like this. So for now, we didn't work on making a system to automatize this. You can load 2 slides into the system, one on the left, one on the right, and start the scan of it.
Okay, thank you, Mickael. And the final question is about the possibility to demo the Orion instrument in my lab. Essentially, people are trying to understand if we do offer a demo of the system.
So the question is about a demo instrument in the lab.
Yes.
So right now we do not engage on in on-site Orion demonstration. However, we do offer proof of concept studies to support our collaborators. So if it helps for the decision-making process, so if people want to discuss that type of demonstration, don't hesitate to contact us so we can have a talk about it. But yeah, we do proof of concept studies with people.
Thank you, Mickael.
You're welcome.
Well, that concludes our webinar for today. Hope you enjoyed it. If we did not get to your question, we will be following up via email in the next couple of days, so do expect that. And have a great rest of your day, great afternoon, and great evening. Thank you.